Welcome back to Chapter 13, Part 2, Using Physical Methods to Control Microorganisms. In this video, we'll focus on the physical ways that we can eliminate or reduce microbes through heat, cold, pressure, drying, radiation, or filtration. By the end of this section, you should be able to compare different physical methods used to control microbial growth and describe how each method works and its practical applications in healthcare, food safety, and laboratory settings. Heat is one of the most effective and widely used methods for killing microbes. It works by denaturing proteins and disrupting cell membranes. And we measure heat effectiveness in two ways. The thermal death point is the lowest temperature that kills all microbes in 10 minutes. The thermal death time is the amount of time needed at certain temperature to kill all microbes. Moist heat, such as steam or an autoclave, is more effective than dry heat because water conducts heat better than air. Boiling, which is a type of moist heat, kills most vegetative cells and viruses, but does not destroy endospores. Some can even survive 20 hours of boiling. Incineration, a type of dry heat, like flaming a loop or using an oven, is dry heat that destroys all organisms, including endospores. Remember that endospores are extremely resistant and require higher temperatures and longer exposure times. And here we have two examples of dry heat. On the left, I see a Bunsen burner and someone is flaming their loop. So this is a type of incineration. We also have these types of incinerators on the right, a bacteria incinerator. And the benefit of having this instead of a flame is that it minimizes aerosolization. It also prevents the use of an open flame in the laboratory. The autoclave, like the one shown here, is the gold standard for sterilization in healthcare and microbiology labs, and it uses pressurized steam at temperatures between 121 to 132 degrees Celsius at very high pressures, so 15 to 20 psi, and this is for at least 20 minutes. This combination kills all vegetative cells, destroys viruses, and even endospores. And to ensure each cycle is actually successful, we'll either use heat-sensitive tape, which changes colors when it reaches the proper temperature, or biological indicators such as Geobacillus stereothermophilus endospores, which confirm true sterilization. So autoclaves are essential for sterilizing surgical instruments, the media that we use in our labs, and glassware in lab as well. Here on the left, we can see an example of the heat-sensitive tape. Here's the tape before we autoclave, and then afterwards, we know that the autoclave has met that minimum temperature, for example, 121 degrees Celsius. On the right comes from our lab manual, so you read this for one of your reading quizzes. This is a biological indicator where we have endospores within an inner vial, and after autoclaving, if you break the vial and release the endospores, if there is fermentation of the medium, you know that it did not reach the required or proper temperature. So the autoclave was a type of moist heat. Pasteurization is also a type of moist heat control, and it uses controlled heat to reduce pathogens in food while preserving flavor and texture. And there are two main methods. HTST, which stands for high temperature short time, is pasteurization at 72 degrees Celsius for 15 seconds. This kills most pathogens, but still requires refrigeration. But UHT, which stands for ultra high temperature, this is 138 degrees Celsius for at least 2 seconds. This kills nearly all microbes and allows the food product to be stored at room temperature, such as the creamers shown here. So if you zoom in and look, you can see UHT, and then it's also here UHT. So these methods prevent spoilage and foodborne disease without damaging the product. So for example, UHT milk can stay unopened for months, whereas HTST milk has to stay refrigerated. So our book shows us the process of pasteurization here and the type of pathogenic organisms that are killed by pasteurization. High temperature, short time, remember there's still microbes remaining, so these foods still have to be refrigerated. In ultra high temperature pasteurization, it's a much higher temperature for a shorter period of time, and as long as they're sealed in airtight conditioners, they can last long without refrigeration. Our next checkpoint question asks, in an autoclave, how are temperatures above boiling achieved? So an autoclave is a sealed chamber that traps steam under high pressure, around 15 to 20 pounds per square inch, or PSI. And increased pressure raises the boiling point of water above 100 degrees Celsius. So it's a lot like a pressure cooker that you might have at home. So this allows the temperatures to increase over 100 degrees Celsius up to 121 to 132 usually, and this is hot enough to kill endospores and destroy all microbes, even viruses. For the second one, how would the onset of spoilage compare between HTST pasteurized and UHT pasteurized milk? So remember, HTST is high temperature short time, 72 degrees Celsius for 15 seconds. This reduces microbes, but still leaves some survivors, and we need to refrigerate these foods because they spoil a little more quickly than UHT. So UHT is 138 degrees Celsius for at least 2 seconds. This kills almost all microbes, so these foods can be stored unrefrigerated for months. They spoil much later. For the third question, why is boiling not used as a sterilization method in a clinical setting? So boiling kills most vegetative cells, but not endospores, like the endospores from Clostridium botulinum, which causes botulism. But some endospores can survive 20 hours or more of boiling. So therefore, boiling doesn't ensure sterility, and that makes it unsafe for surgical or lab use. Cold temperatures don't kill microbes, but they slow their metabolism and growth. So refrigeration and freezing, these are static, right? They inhibit growth, but they do not kill. Refrigeration, which is between 0 to 7 degrees Celsius, like our fridges at home, they keep food and medical supplies fresh. So these are bacteriostatic, but not bactericidal. Freezing, which is usually less than 2 degrees Celsius, halts metabolism and can kill some microbes, but many survive and can grow again when thawed. Ultra low freezing, which is minus 70 degrees Celsius or below, is used for preserving microbial cultures or vaccines long term, and they show us a freezer here. And we also have liquid nitrogen tanks that are shown down here for long-term storage or transport. So overall, refrigeration and freezing preserves but does not sterilize. So does placing food in a refrigerator kill bacteria on the food? The answer is no. So refrigeration slows metabolism and growth but does not kill most microbes. We have to worry about psychrophiles. Remember, psychrophiles are cold-loving bacteria. They can still grow slowly at low temperatures. So refrigeration mainly preserves food by delaying spoilage. So in our book, in this section, they have a case study on a potluck where people ate home canned potatoes, and about 24 hours later, they have signs and symptoms of botulism. So Clostridium botulinum, remember, is endospore producing. And if endospores are ingested and they begin to germinate, the germination process of becoming active cells again will usually result in the production of toxins like botulism toxin. So to kill endospores, destroy endospores, it requires a minimum temperature of 116 degrees Celsius. That's a lot higher than what we can reach at home. The boiling point of water is 100 degrees Celsius. So people usually recommend having a pressure canner to reach temperatures above 100 degrees Celsius. Otherwise, the CDC recommends boiling home canned foods for about 10 minutes to denature botulism toxin before you eat the food. Additional physical methods to control microbial growth include pressure, desiccation, and high osmotic pressure. So high pressure processing is a modern method used in the food industry to kill vegetative cells and destroy viruses without changing the texture or taste of food. But endospores can still survive at high pressures. Desiccation or drying, also known as dehydration, removes water, and microbes can't metabolize without water. So foods like jerky, powdered milk, or dried fruits use this principle. Lyophilization or freeze drying combines freezing and vacuum drying, and it's used to preserve vaccines, bacterial cultures, or foods by halting growth and maintaining structure. But drying is static, so metabolism or growth will often restart if you re-add water. Osmotic pressure can also inhibit growth. So if you have high sugar or high salt added to your food, this draws water out of microbial cells, right? This would make the environment hypertonic, and when cells are placed into a hypertonic environment like high salt or sugar, they will lose water, they'll be dehydrated, and this might kill some cells. So that's why salted fish, jams and jelly, or honey can resist spoilage. Although some microbes, in particular molds and yeast, unfortunately, can be tolerant of desiccation, and sometimes they can still spoil these types of foods. Here we have several examples of desiccation, lyophilization, and high osmotic pressure. So desiccation is drying out. So dried apricots or these raisins, those are examples of desiccation. And lyophilization is freeze drying. So I had to pull these pictures from the internet. So if you have these freeze-dried strawberries or astronaut ice cream, these are examples of lyophilization or freeze drying, and you might see these also in your cereal. Even the cereals with the sugary marshmallows, like I think Lucky Charms, those are freeze-dried as well. So since they don't have any water in these freeze-dried items, you'll see they're usually crunchy compared to this more chewy dried apricot. We also have dried foods, and some of these have been salted. So that adds on osmotic pressure, that osmotic pressure element, like jams and jellies. And in high osmotic pressure, these hypertonic conditions, cells will lose water. So that's supposed to be a bacterial cell, for instance. Kind of looks like a donut to me, though. But you can see water is leaving the cell, causing sometimes cell death. So how does the addition of salt or sugar to food affect its water activity? High concentrations of salt or sugar draw water out of microbial cells. This is through osmosis, because this creates a hypertonic environment and dehydrates the cells, which stops their growth. So examples are what we just saw, like salted fish, jams or jellies, and honey. These resist spoilage because water activity of bacterial cells is low. Radiation can also control microbial growth by damaging microbial DNA, and there are two main types we've discussed before. Ionizing radiation through X-rays or gamma rays can create double-strand DNA breaks. So ionizing radiation can deeply penetrate surfaces as well as packaging, so we can use it to sterilize medical supplies, gloves, or food products. Non-ionizing or UV light, this type of radiation causes thymine dimers in DNA, and that prevents DNA replication. So this is used for disinfecting air, surfaces, and water. Ionizing radiation is more powerful and penetrative, while UV is used really only for direct surface exposure because the UV light cannot penetrate plastic or glass. If you use foods that are treated with gamma radiation, they have to be clearly labeled with a symbol I'll show you shortly called the Radura symbol, and this indicates safe, regulated irradiation. Here we have a double-stranded DNA segment with DNA thymine dimers caused by exposure to UV radiation. On the right, we see that a lot of our biological safety cabinets have UV light built in order to sterilize the equipment underneath. What are two advantages of ionizing radiation as a sterilization method? So one is deep penetration. X-rays and gamma rays can pass through packaging. They're also highly effective. They cause double-strand DNA breaks, killing even endospores. So it's useful for heat-sensitive medical supplies and food preservation. For the second question they ask, how does the effectiveness of ionizing radiation compare with that of non-ionizing radiation? So ionizing radiation is stronger. X-rays and gamma rays penetrate deeply and sterilize completely, whereas non-ionizing radiation like UV light has pretty shallow penetration. So it's best for surface disinfection or disinfection of the air. And UV light, remember, forms or causes thymine dimers in DNA, but doesn't kill microbes inside materials because it can't penetrate packaging. Although gamma radiation is a proven method of eliminating potentially harmful microbes from food, there's very low buy-in by the public, usually because of poor understanding of the principles of radiation. So the food does not directly contact the radioactive material, right? It doesn't become radioactive food. There's no risk for exposure to radioactive material through eating gamma irradiated foods, but it's still hard for the general public to understand if they're not scientists. Any kind of gamma irradiated food has to be clearly labeled and will display this symbol. This is called the Radura symbol. So you might encounter the Radura symbol on the foods you buy. I've seen them most recently on a box of cookies at Whole Foods. I can't remember what the cookies were called, but I remember the box had a sentence on it. It said something like, the best cookies in the world. So I'll have to go back and maybe buy those. Here we have strawberries that have been irradiated versus those that have not. You can see the ones that have been treated last much longer because the microbes on their surface have been destroyed. Sonication and filtration are also physical methods of control. Sonication uses high-frequency sound waves to disrupt cells, so it's useful for breaking up biofilms or lysing bacterial samples in the lab. Filtration physically removes microbes instead of killing them. So we might use membrane filters that have a pore size of about 0.2 micrometers to trap bacteria or larger particles from the liquids. And this is great for sterilizing anything that might be heat sensitive, like antibiotics or serum. HEPA filters remove particles that are greater than or equal to 3 micrometers from the air, and these are essential in hospital ventilation systems as well as our biological safety cabinets. And HEPA filters maintain sterile airflow in our labs and prevent airborne pathogen exposure. Here we have two examples of membrane filters. Here's an apparatus. Here's the membrane filter with that 0.2 micrometer size, that pore size. And we have a vacuum here attached. So the vacuum will draw the liquid down through the filter. And we work with these, for instance, in our majors biology class, where we're working with bacteriophage versus bacteria. The bacteria get trapped above the filter because they're bigger than the 0.2 micrometer size, whereas the viruses can go through, and we can isolate the viruses that way, those phage. If we're working with small volumes, we'll look at syringes. So we'll have usually very small membrane filters that attach to a syringe for small volume work. Our next checkpoint asks, would membrane filtration with a 0.2 micrometer filter likely remove viruses from a solution? The answer is no. So most viruses are smaller than 0.2 micrometers, so they will pass through the filter. The 0.2 micrometer or micron filtration removes bacteria and fungi, but not viruses. If we wanted to remove viruses, we would have to use something called nanofiltration, or we could use chemical or physical sterilization. For the second question, name at least 2 common uses of HEPA filtration in clinical or lab settings. So one could be in our biological safety cabinets to protect the lab workers from aerosols, especially if they're pathogens. And the second could be hospital isolation rooms to prevent airborne transmission like tuberculosis. HEPA filters are also used in operating rooms and clean rooms for sterile airflow. And at the end of this section in our textbook, they gave us a big table of all of the different physical methods of control. So let me zoom in on this. Examples that we've covered so far include heat. So we had moist heat like boiling, autoclaving, or pasteurization, and dry heat like dry heat oven or incineration. We had cold temperatures to inhibit growth. So this is more static. And then high pressure examples. We also had desiccation methods where we're drying out or removing water to lower water activity to inhibit metabolism, but these are often static because if you reintroduce water, the cells might metabolize once more. And then we just looked at radiation as well. We had ionizing or non-ionizing radiation. We also have sonication where you're exposing the substance to ultrasonic waves. You might have seen videos online of people cleaning their jewelry using these high vibrations. These cause lysis of cells. And we just saw filtration as well. We had HEPA filters with 0.3 micrometer sizes or membrane filters with 0.2 micrometer sizes. And our 13.2 learning objective really asks us about the tables that we just saw. So we want to understand and compare all of these different physical methods of controlling microbial growth: heating, refrigeration, freezing, high pressure, desiccation, lyophilization, irradiation, and filtration. So what I did here is to provide an empty table, and in the next slide, you can see the answer key. All right, and here is the key. This is kind of an abbreviated key compared to the previous table and slides, but it's nice to have an overall general picture of each of these physical control methods. And that takes us to the end of part 2. In the next video, we'll cover section 13.3, using chemicals to control microorganisms.